So we investigated that, and Goldman, Babitsky, Saussure, Valverde, and Guanan Chen shows that Bacillus produces an anti-trap protein when tRNA trip is on charge. An anti-trap protein binds the trap and prevents the trap from binding to RNA. And you, of course, wondered how was AT synthesis regulated. And what Saussure and Marino found is that in the AT operon, right downstream from the upstream from the promoter, there's a what was called a T-box regulator. And this is a common regulatory mechanism in Bacillus. So there's approximately 200 genes involved in amino acid metabolism are regulated by T-boxes, and these T-boxes bind an uncharged tRNA. And in this particular case, what we're looking at is uncharged tRNA trip. It binds by codon-anticodon pairing to this loop region with a trip codon. It also binds the acceptor end that doesn't have amino acid on it and stabilizes this anti-terminator. Anti-terminator and terminator are alternatives. If the tRNA is uncharged, you stabilize this with no termination. And if there's only charged tRNA trip, the anti-terminator is not stabilized, and you form the terminator and transcription is terminated. Okay? Yes, we show that the AT operon is transcriptionally regulated by an uncharged tRNA trip. And Guanan Chen investigated this, and he showed that if you take this temperature-sensitive mutant and you delete the AT operon, so there's no AT protein, lo and behold, the operon expression is low, confirming that the AT is working, that the explanation is correct. But as usual, when you do science, you always think of one additional experiment to confirm your conclusion. And you do it, and sometimes the answer is opposite of what you expect. And that occurred here. What we did was just delete the T-box. If you delete the T-box, you should make plenty of AT, you should inactivate trap, but that didn't happen. T-box deletion increases the entropy only slightly. Why? Why did that happen? Okay. And of course, I assigned Guanan Chen this question problem, and he discovered that downstream, immediately downstream from the T-box leader, there's a short coding region that codes for 10 residue peptide. And this peptide has 3 adjacent tryptophan residues, and the stop codon is only 6 nucleotides for the Shine-Lugano region. Okay? So what he showed was that when translation is complete and the ribosome reaches the stop codon, it masks the Shine-Lugano and you don't make the AT protein. But if the cell is deficient in charged tRNA trip, the ribosome translating this message stalls at one of the trip codons that exposes the Shine-Lugano region, and you do make the AT protein. So very sophisticated, both transcriptional and translational regulation of AT synthesis. So these are the events that occur that we deduced. With a sufficient charge tRNA trip, transcription terminates in the T-box leader region. When there's a mild charge tRNA trip deficiency, the ribosome reaching the stop codon inhibits AT synthesis. Okay? It reaches the stop codon, inhibits AT synthesis. When the severe charge tRNA trip deficiency, ribosome translating leader peptide protein stalls an internal trip codon, exposing the Shine-Lugano, and so the AT protein is synthesized. And that's the way it occurs. Now, when trip is present, but tRNA trip is largely uncharged, AT is produced, AT binds to an activating trap. In activating trap, the anti-terminator forms and persists, terminator cannot form, and RNA polymerase continues transcription. So that's the way it works. Now, this is a hypothetical model produced by Angela Balbuzzi in my lab. We thought AT bound to the surface of trap, prevented trap from binding RNA, and some confirmatory experiments were produced in collaboration with Golding and Babitsky, who has left my lab. This shows two subunits of the trap protein with a tryptophan binding site, and the work of Golding and Babitsky had identified three basic residues that are required for RNA binding, these three, and three new residues that require tryptophan binding. And in a collaborative study, we showed that you change any one of these residues, not only does trap not bind to RNA, but trap no longer binds the AT protein. So the AT protein is uniquely designed to recognize the surface of the trap protein that's formed when trap is activated. The structure of AT has been solved in trimer. It's an equilibrium between trimer and 12mer, but the structure of the AT trap protein is not known. Okay, so running out of time, so this will summarize the interesting findings in years of studies with trypophan regulation in the cellulose. We identified two regulatory proteins, trap and AT, and two signals, tryptophan and tRNA trip, that's recognized. When tryptophan concentration is high, trap is active, transcription is low. When tryptophan concentration is low, trap is inactive, transcription of the RNA is high. When the tRNA trip concentration is, when tRNA trip is mostly uncharged, AT is synthesized, AT inactivates trap, transcription is high. When tRNA trip is mostly charged, AT is absent, under these conditions tryptophan is high, trap is active, and transcription is low. Complicating the sophistication is this final point that tRNA synthase, which charges tryptophan on the tRNA, is regulated by T-box, by tRNA. So there's another level of regulation influencing everything on this ride. So when tRNA trip concentration, tRNA trip is largely uncharged, tRNA synthase level is high, and when it's charged, tRNA trip level is low. I'll show two more slides. Okay. Okay. This, of course, once you understand some phenomena, you begin to ask questions about what about other organisms. And I convinced one of my former postdocs, Rhee, who become a genome specialist, to actually look at all gram-positive as sequences that have been a bacterial sequence that completely sequenced, and just see if there's an arrow superoperon in any other organism. And what he found is that there are several organisms with arrow superoperons. They also have trap binding sites before trip B, and they also produce a trap protein. But surprising finding is that only one of these, the closest related to bacillus, produces an AT protein. So we don't know how these others recognize tRNA trip. And what was also surprising from these studies is that most of the other gram-positive bacteria had all the trip genes in a single operon, shown here, not in the superoperon. And furthermore, we were surprised to find that they were regulated by T-boxes and by tandem T-boxes, recognizing uncharged tRNA trip. Why have tandem T-boxes? Well, the advantage of the tandem T-box versus a single T-box is each of these allows 10-fold regulation. If you have two of them in sequence, you have 100-fold regulation. So it remains to be seen whether tryptophan is recognized in the regulation of any of these organisms. So I don't have time to describe our work in glycoformins performed by our last postdoc, or our last project that we worked on was regulation of the tryptophanase degradative operon which is very exciting and very interesting, because those studies show that a nascent leader peptide can instruct a translating ribosome to create a binding site for a specific molecule that regulates translation termination or elongation. So you can regulate the ribosome by the amino acid sequence.